which to make organisms that store and retrieve more information. So here it is. Here's the genetic alphabet, and I think everyone's familiar with it. What's remarkable about the genetic alphabet, at least to a chemist, is how simple it is. It's just a linear polymer of 4 different nucleotides attached in a simple way. But of course, what makes DNA so remarkable and unique is that it has a specific molecular recognition that underlies both its stable pairing as well as its replication. And that, of course, is that A always pairs with A always pairs with T, and G always pairs with C. And so all of the diversity that's existed in life, best we can tell ever, everything that you see around you is encoded within that 4-letter genetic alphabet. And the idea from the beginning was to try to create a 5th and a 6th nucleotide that stably pair together and that are recognized by all the machinery of a cell required to store and retrieve that information. So here's sort of an overview of what would be needed for such a project, and we call these organisms semi-synthetic organism because the idea would be that they would have a synthetic, a man-made part in their guts, in the very fundamental aspects of their information storage, and that is with that 5th and 6th nucleotide. So if one wants to do that, you have to be concerned. You have to have some sort of an unnatural nucleotide, which I'm calling Y here. It has to be in some sort of an episome or a chromosome or something. And you have to have a DNA polymerase that, when it encounters that nucleotide, has available to it triphosphates of the partner nucleotide, such that efficient replication and high fidelity replication occurs to propagate and store that information. And then downstream, you have to have the same story for RNA polymerases. You have to have, when an RNA polymerase encounters the unnatural nucleotide going in either direction, the triphosphates have to be available such that RNA can be efficiently synthesized to produce both mRNA for the ribosome decoding, with the tRNAs with cognate unnatural anticodons that are charged by unnatural amino acids, thus enabling the incorporation of non-canonical amino acids into proteins with, in that way, hopefully expand the function of proteins, which are receiving all sorts of interest in terms of, for example, protein therapeutics. Now, from the beginning, I want to mention that the orthogonal pair of tRNA synthetase that one uses for this, it has to be orthogonal, meaning it can't interact with the natural system. And a couple, 2 good ones have been developed, both from Archaea. They were developed to read stop codons. This is the famous AMBER suppression method. And we planned on using those very same orthogonal translation systems, except instead of recoding the tRNAs to recognize an AMBER stop codon, which has its limitations because it limits the number of unnaturals you can incorporate. It competes with release factor. It's challenging. So instead of recoding those tRNAs for AMBER stops, we would recode them with our unnaturals for to have unnatural anticodons, and therefore make virtually an unlimited set of new codons that one could harness for information storage and retrieval. So towards this goal, at the very beginning, we spent a lot of time thinking about what sort of properties we would try to harness in these to develop these 5th and 6th nucleotides that form an unnatural base pair. So for a variety of reasons, we chose to go after hydrophobic pairs, in part because I think it was just a neat question, because what we were trying to do here was develop a 5th and 6th nucleotide, and baked into that question, whether we could or not, was the question of whether the 4 natural nucleotides that nature uses are in some way absolutely unique and could not be altered or expanded on in any way, and a chemist certainly couldn't come in and play with these analogs and develop a new base pair that functions within and alongside the natural system. So I think that was an interesting question. But the idea of using hydrophobicity, which underlies protein folding, a good part of it, and is a very strong force. Not only did we want to use that because we thought it was a strong force, but I think it was an interesting question to ask. Not only could we develop unnatural base pairs that are very different from those used by nature, but could we in fact use a force that was completely unlike that used in nature? So we weren't going to try to harness complementary hydrogen bonding. Instead, we wanted to develop complementary packing and hydrophobic forces. And so I'm showing here what became sort of known as our 1st generation analogs. And there's a couple things I want to say about them. First of all, I'm only showing the nucleobase part. Some of them not even basic, but I'll refer to them as bases just for simplicity. I'm not showing the ribose or the phosphate. I'm only showing the nucleobase analog. So a couple things I want to say about them. First of all, is that obviously for reasons that I just described, they're all hydrophobic. And secondly, I want to mention that there's a lot of them. And that was a very important point because we didn't want to take the sort of, I don't know what you might call the traditional biochemical approach, where we very carefully design something and then see if it works, because I didn't think that we knew enough about polymerases and even about duplex DNA stability to be able to design something. So we took very much a medicinal chemistry-like approach. I've always been inspired and impressed with medicinal chemistry and how they develop drugs, and we tried to apply the same method to develop our base pair. And that means that we synthesized analogs, we characterized them with assays to develop structure-activity relationships, and then made modifications to fine-tune those SARs, and then used that data to feed back into the design process to continue optimization. So this was a lot of analogs that we made. These eventually led to sort of a second-generation analog with SAR that suggested that pairs should be smaller and not quite as large. And so we examined lots of analogs, again, looking at sort of halogen substitution, shape modification, the introduction of other functionality. And these became known as sort of our second generation analog. We were making a lot of progress, and then around 2006, we ran a high throughput screen where we actually characterized all of the pairs against each other. And that was really important for us because we identified our first pair that was important, that was efficiently replicated by a DNA polymerase, and that's this pair that we call 5,6 for 5-methyl sulfur isocarbyl styrol. That's as IUPAC as I'm going to get, and that was a first generation analog. And that paired well with this second of the methyl methoxy series that we call MMO2, which was a second generation analog. That very quickly reinvigorated our design effort, and we came up with this 5,6 NAM pair by expanding MMO2 out with this aromatic ring positioned here. And it became a much better partner for 5,6 than was MMO2. And this was a very important pair for us because it was the first pair that we actually could replicate in a PCR reaction with high fidelity. So we were very excited by that. And I'm going to talk a lot about that in the next 10 minutes or so. But the efforts continued to optimize the pair, and we came up with this analog that we call TPT3 by contracting the distal ring and adding a heteroatom to it. And it's a better partner for NAM than is 5,6. And so don't forget TPT3. I'll return to that in a little. But a lot of efforts, we only more recently discovered TPT3, so a lot of our efforts focused on this pair. The SAR the assays that drove that SAR were largely steady state kinetics, and the problem with this new pair was that it's replicated so efficiently that under steady state conditions, it's replicated as efficiently as a natural base pair. Now that's exciting, but it's also problematic because it just means that we've optimized the rate of synthesis sufficiently that the product formation is now limited by a non-chemistry step, a pre-chemistry conformational change in the polymerase that's known to limit the rate limit the synthesis of a natural base pair. So what that means is that we had sort of tricked the polymerases into making this pair efficient enough that they thought they were natural, but it also prevented us from using steady state kinetics to drive further optimization. So we developed another assay. It's a PCR assay where we simply take a PCR amplicon of an oligonucleotide containing the unnatural base pair, we amplify it, and then we simply give it to the sequencing facility, and they run a standard sequencing reaction on this. And for me, this was really exciting because in a lot of ways, it felt like it was the first time that we ever saw the unnatural base pair. So here's just a normal sequencing chromatogram, and you get this abrupt termination at the position of the unnatural base pair, and then a much smaller amplitude read afterwards. And so this small amplitude read almost certainly results from the sequencing reaction itself because the unnatural triphosphates are not present. But this gives you this ratio, once normalized, it gives you a minimum fidelity once you divide by the amplification level. And so we started to use this as an assay. And so we looked at the unnatural base pair. This is now 5,6 NAM again. And we put it into a bunch of different sequences. I'm only showing the immediate flanking sequence here. These were 150 mer oligonucleotides. They were amplified they were amplified 10 to the 12 fold. So that's huge amplification because if there was mispairing, we wanted to make sure we gave it a chance if it was more efficient to dominate the population. But the efficiencies remained very high, and most importantly, the fidelities remained very high. So we were very encouraged by that. We could even replicate 2 unnatural base pairs in a row. We even made random libraries where the fidelity also remained very high, suggesting that there were no natural sequence context within the unnatural base pair where it would be lost efficiently. So we were very excited about that. And we considered then going on to our long-term goal of expanding the alphabet. But we had structural studies that showed something weird. I very briefly want to describe this. What it showed was that the pairs weren't doing this. They weren't edge-on pairing. They were cross-strand intercalating. Everyone knows that DNA polymerases are supposed to select for correct Watson-Crick pairs. It doesn't matter whether it's a GC, CG, TA, or AT. They all form a conserved structure which the polymerase is supposed to recognize. So the way this works is a polymerase, once it binds a template triphosphate, once it binds the correct triphosphate, the fingers domain closes down over the palm and thumb domain and forms a tight closed complex that's supposed to rigorously recognize the Watson-Crick-like structure. But our pair is not forming a Watson-Crick-like structure, edge-on structure. It's forming this cross-strand intercalated structure as I'm showing here. So we had two questions. Number one, was formation of the unnatural base pair sufficient to drive that same large conformational change? And the number two, if it was, what the heck is it recognizing? Because our pair doesn't look anything like a Watson-Crick pair. So we collaborated with Andy Marks and his grad student Karen Betts at the University of Constance in Germany. And I'm going to just very briefly describe what we found. There's a variety of crystal structures, and here's the binary complex showing the polymerase. The only part of the polymerase I'm showing is the O and the O1 helix. That's the base of that fingers domain that I mentioned. Here's the primer and the template with our analog flipped out, stuck into a hydrophobic pocket at the base of the O helix. When you add the correct triphosphate, that unnatural in the template flips back into the developing duplex, and you get this large conformational change of the polymerase. To show you that conformational change, here's an overlay of the binary and the ternary complex, and there's the conformational change. To convince you that it's exactly the same as the conformational change induced by a natural base pair, here's an overlay of the ternary complex synthesizing our pair with the complex synthesizing a GC pair. So you can see at the secondary structural level, it's virtually identical. At the side chain level, and even the bound waters and magnesium ion level, it's virtually identical. So the answer to the first question is very much that it does induce the same conformational change. So the second question, what is it recognizing? And this was really exciting to us, because what we found was that when the unnatural flips back into the developing duplex, it does so in a way that forms the correct Watson-Crick-like planar structure. So the unnatural base pair is not being replicated in some weird intercalated structure. It does form a correct edge-on structure, but only during replication after it then translocates out of the polymerase active site, it recross-strand intercalates. And this is almost the ability to adopt the in-plane structure for replication is almost certainly what we optimized for years in that SAR effort. So having a pair that we believed replicated well, replicated in a sequence-independent manner, and we even believed that we understood the structural mechanism, we then initiated efforts to actually deploy the system in the living organism. Now, the first step one has to be concerned about is the step I glossed over initially, and that is, how do you get the unnatural triphosphates in? And for any students out there, I really like to illustrate what I think was a great example of interdisciplinary science. And what I mean by that, and what I think interdisciplinary science often is it just means you read other people's literature. And so what we found out, there were a large number of people, a few people who were working on dedicated nucleoside triphosphate transporters that transport triphosphates from host organisms into lipid bilayers into organelles or obligate intracellular bacteria. And a variety had been characterized. We rode around different labs across the world, got a bunch of them and tried them, and found one that worked really well. It was from an algae species, and it's called PTNTT2. It assembles in the periplasm. So the way the system works is we add the triphosphates to the media, they diffuse in through the outer membrane porins, and then are actively transported into the cytoplasm by virtue of the nucleoside triphosphate transporter. So to see if the whole system worked, we put together a couple of plasmids, one we called the accessory plasmid, and all it had right now was just the transporter that I just mentioned. But we also constructed what we called a PIMF plasmid. So those of you who think about plasmids, this is just PUC19. This is just PUC19 where a single nucleotide, shown right here, a single pair of nucleotides has been changed from an AT to a XY. And XY, remember this TP53 analog that I mentioned earlier? We now knew that it replicated in a PCR reaction better than 5,6-NAM. So the in vitro plasmid where we constructed the unnatural base pair was made with this pair. The experiment I'm about to describe was made with this pair. And so this is going to come back to be important in a few seconds. So here's the actual experiment. So we took the plasmid, it has the unnatural base pair, the information plasmid PIMP, and we transformed bacteria that already harbored the accessory plasmid, grew them for periods of time, in the presence of the triphosphates in the media, and then recovered the plasmids and PCR amplified the flanking region. And we did so in a manner such that one of the unnatural nucleotides was by it and tagged. And the reason we did that is that allowed us to separate the amplification products that retained the unnatural base pair and those that did not, telling us what the status of the original plasmid was. So here's the data. If you just use PUC19, meaning there is no unnatural base pair, then it doesn't matter if you add the triphosphate or not, you don't get a biot and shift. So this is just a gel showing the migration of this amplicon. If you have the unnatural base pair in the template and you don't add the triphosphate, then you get no shift. If you have the unnatural base pair in the template and you add the triphosphate, and you provide for the uptake by induction of the transporter by IPTG, you see you get a shift. And so that reflects the retention of the unnatural base pair in the plasmid. So we were really excited. We thought that demonstrated retention of the unnatural base pair in a living organism. We wanted to show this separately in a different assay. So here's that sequencing assay that I described. Here's all the controls, plus or minus base pair, plus minus induction of the transporter. And you see that the sequencing chromatogram abruptly terminates right at the position of the unnatural base pair. We can sequence this. We know it hasn't moved around. So as a last assay, we actually took the plasmid, digested it, and sent it to Ivan Correa at New England Biolabs with his group, where they took the plasmid, they digested it down to individual nucleosides, and shot those on an LC-MS assay. And here's the content of that plasmid. Here's the C, G, and A content. T, due to ion suppression, doesn't fly. This was expected in their assay. But right out here, right where it should be, at roughly 1 per plasmid, was this unnatural nucleotide. And remember, we've constructed the plasmid in vitro with TPT3. The only place 5,6 was ever provided was as a triphosphate to the media, demonstrating in vivo replication. So here they are. Here are the semi-synthetic organisms. We thought this, and I still think this is really exciting. Every living organism ever before this has had 4 base pairs, four letters, 2 base pairs, and these organisms are growing and dividing with six letters and 3 base pairs. So in order to move on and try to develop fully functional SSOs that retrieve that information, we synthesized the ribo analogs. We actually knew during the development phase that the analogs were both, not only were they replicated well, they were transcribed well. And so that we also knew the transporter imported them. So now we relied on replication of the plasmid for retention of that base pair, transcription into tRNA and mRNA in vivo, and then translation at the ribosome to produce proteins with unnatural content. Now I'm going to reduce a bunch of work to one slide, but there it is. So we took a GFP gene and put the unnatural base pair in the middle, and then we provided the one of those orthogonal translation systems that I mentioned. In this case is from the archaeal species Mizet. And so that's producing the tRNA and the tRNA synthetase required to charge that tRNA, which is then recoded with an unnatural codon that anticodon that recognizes the installed codon. Now down here I showed the different codons that we explored. In each case, they were of course provided with their tRNA with the cognate unnatural anticodon. And the amino acid that we incorporated was an azidolycine analog. And the reason we did that is that allows us to show, to demonstrate incorporation. We of course use mass spec, but we also wanted a facile assay to demonstrate incorporation. And so since it has an azido group, we used click chemistry to attach a dye. And so the dye, we found out, very conveniently induces a gel shift. So here's a control reaction where we're using amber suppression to incorporate the unnatural. And so here's the control of just the GFP itself. And so you see the shift and a very high fidelity shift. But here are all the unnatural codons. In fact, here are 11 unnatural codons. These are just shown on a different plot because they were actually technically done in a separate experiment. These are all done many times, and representative gels are shown, but these were not done side by side, so I did want to separate them. But here you see all these different codons. Maybe this codon, GXG codon, decoded by CYC, where X is NAM and Y is now TPT3 in the anticodon. You can see that has a little lower fidelity, but the rest of these are very high fidelity. We also showed we simply selected 3 of them and showed that they were mutually orthogonal, meaning that you only got GFP fluorescence when you had a codon with its correct anticodon, so that you didn't get any crossing between the codon anticodon pairs. And of course, what that allowed us to do was to prepare proteins that were double tagged by simultaneously decoding 2 unnatural codons. And so here is a GXT and an AXC encoded within the GFP, and we're now providing 2 orthogonal translation systems from Mazea and from Jinashi. Those are the only 2 that are well validated and well known. Here's the growth relative to an amber ochre suppression. And you can see that with amber ochre taking up 2 of the 3 stop codons, the cells really don't like it. And they're not growing very well, and you don't produce much protein. And you can see that the protein production in our system is much higher. In fact, it's almost as high as decoding a single, which really I think starts to reflect the utility of having these truly new codons. And so here, using copper chemistry and strain-associated click chemistry, we showed that we indeed incorporated 2 by showing this double shift. And then here's the mass spec showing high-fidelity incorporation of both unnatural both non-canonical amino acids. To really demonstrate the power, we decoded 3 unnatural codons at the same simultaneously, which of course is not possible with amber, so we don't have a control to show, but the protein expression remains very good, decoding all 3. I want to mention now that we did have to use the E. coli tRNA seer tRNA, which was recoded for one of the anticodons, for one of the codons, because there aren't 3 good orthogonal systems. But nonetheless, it is decoding the unnatural codon, and you see you get protein production, and the mass spec shows very high fidelity. A couple last things that I wanted to mention is that we are still optimizing the chemistry underlying the unnatural base pairs. And what we found is this pair, which we call CNMO, is a derivative of MMO2, which replicates at the DNA level better. And in terms of the ribo pairs that we use, this NAM triphosphate is still optimal, but we found that taking TPT3 and adding this nitrogen atom here significantly improves its behavior as a ribotriphosphate during transcription. So there's a lot of data here. I don't want to show it to you all. I just want to show you one. I want to draw your attention to one part, and that is, in this case right here, we're decoding 3 unnatural codons that are separated by a natural. So this is GFP with an unnatural at 149, a natural at 150, an unnatural at 151, a natural at 152, and another unnatural at 153. And with this original system that I just described, you can see that that's a very difficult to replicate sequence. So this is a protein shift. So this is not making protein. And we know that the problems arise at the replication level because we get about equal parts protein with all 3 non-canonicals incorporated, 2 non-canonicals and 1 having been lost, 1 non-canonical with 3 lost, and even some that lost all 3. It's a very difficult sequence. But with this new system shown here, you can see that there's the protein now prepped. It virtually all has the triple label with very little second case where one of the non-canonicals has been lost and virtually no further loss. So we're very excited about that. I very briefly want to mention that the system works in eukaryotes. These are CHO cells. We have not yet got the transporter system working in a eukaryote, but so what we're doing here is we're transfecting CHO cells with mRNA and tRNA that already have the cognate natural, unnatural codon and anticodons, and you can see that this is an amber control. You can see that you get nice, maybe this is most illustrated here in the TAMRA channel. You can see you get nice unnatural incorporation in all cases. And we examined 5 codon anticodon pairs there. I very briefly want to mention in the last 2 minutes that I have is that there was a company called Synthorex, stands for synthetic organism with an X on the end. 2014, that by 2017 they had a commercial product. Well, they had a ongoing clinical trial. And they've taken, without giving you too much detail, a IL-2 therapeutic, which is a cytokine which has a lot of promise, but it has a broad range of targets that it hits, and it's also toxic. And so there was an idea as to how we could make it not toxic by selectively positioning an unnatural amino acid. And you can say that protein, which we call Thor707, and this is in looking at tumor volume in a mouse. You can see that as a single agent, it dramatically reduces tumor volume. And in actually looking at the much more stringent survival curves for the mouse, we were looking at it in combination with an anti-PD-1 inhibitor, which I'm sure you've heard about. They've all the rage in anti-chemotherapy in immunotherapy. And you can see that these are very difficult to treat tumors. Here's the untreated sample. Here's the sample treated with only the checkpoint inhibitor, the anti-PD-1 antibody. Here's the system treated only with our agent, which is actually better than the PD-1 antibody. And in combination therapy, it was most exciting because 5 of the 14 mice were actually cured. Really exciting. At the beginning of this year, as I mentioned at the beginning of the talk, Synthorx was acquired by Sanofi. So I'm now a Sanofi employee, and I'm very, very excited about helping to implement this technology in a much broader range of applications. And I look forward to moving forward with Sanofi on that project. And so with that, it just is left to thank the students who worked on this project at Scripps, the Synthorx group, which is now Sanofi. And I think I mentioned these collaborations and these funding agencies. Of course, I'd like to thank for their funding. And so with that, I'd like to thank you for your attention.
Thank you so much, Dr. Romsberg, and congratulations on the acquisition. Super exciting. And as I mentioned before, we'll hold questions till the end of the webinar. So if you have any, feel free to type them in, but we'll address any questions at the end of the event. And to move along, I'd love to have Dr. David Baker, if he's available, proceed with his presentation.
Yeah, okay, I will share my screen now.
Awesome.
And
thank you.
Let's see. Let's see, view slides. Can you see my screen? Can you see the slide?
Yes.
So we see the slide, but only well, someone else maybe. If it looks okay to you, let me know via chat or otherwise. But David, it looks like we're seeing like the not the presentation mode of the
You were
screen.
not seeing. Okay, let's see.
We see your screen, but yeah, we see. There you go. Oh, you had it.
Okay, let's see. Let me just. Sometimes that happens. Sorry about that. Maybe I'll just make the slide as big as possible. What do you see now?
Yep. I think we may have. I think the top may be cut off a bit, but you were okay.
Let's see. Sorry about that. I don't really understand why that happens sometimes. Is it okay now?
You're almost there. If you scroll up, I can tell you when you're hitting the.
Okay, it's funny because it's not giving me the
presentation option. So if you hit the bottom,
I can present. I guess I could try going into presenter mode and then try screen sharing again. I don't know if that works better. How about this?
Yep, and then scroll up just a little bit, and I think we'll get the top of the.
Yeah, it's not giving me the option of that.
Oh,
Sorry
option?
about this.
Oh, no worries.
Let's see. Let me try going into presenter mode and then sharing. Actually, no, I don't know if that will work. That's strange. This usually works for me. Sorry about this.
No problem.
Let's see. Maybe if I stop sharing and.
Someone said it. Alexandra says it looks fine to her. So it may be just me. If you want to give it a go, and if people can't see, just
should I try
let me know.
going into presenter mode one more time?
Why don't you try it just in case, because I imagine maybe some people aren't seeing.
I think it's going to be easier to see. How about now? Can you see presenter mode?
I'm still seeing the same.
Okay. All right. Sorry about that.
Just
we'll just go with that then.
Yeah.
Yeah, I don't know why that's happening. Okay, so I will get started then. Thank you very much for the invitation, and
sure.
I'm going to give you an overview of what is now possible with de novo protein design. So first, I'll introduce the problem. So I think, as most of you know, proteins fold to their lowest free energy states. So if you are predicting protein structure from sequence, you have to search for the lowest energy state of the protein chain. In the protein design problem, instead, we're making up brand new proteins on the computer, and the design problem is to identify amino acid sequences which will fold to those structures. So if we want to we first make up a brand new protein structure, and then we search on the computer for an amino acid sequence which has as its lowest energy state that structure. And we do the energy calculations and the searching in sequence and structure space with a computer program called Rosetta we've been developing over the years. So I'm going to focus today on de novo protein design. And what this means is designing proteins completely from scratch, not starting from a naturally occurring protein. So in this depiction, you see this rectangle. Can you see my mouse actually?
I can.
Okay, cool. Okay, so this depicts a sequence space. It's very, very large. So with 20 amino acid possibilities per position, this is even without Floyd's elegant expansion. For a 100 residue protein, you have 20 to the 100th possible sequences, which is on the order of 10 to the 130th, a truly astronomical number. This is many, many, many orders of magnitude more than what exists in nature. And in de novo protein design, we're searching this full space. Most protein engineering has involved making small modifications to naturally occurring proteins, which we kind of affectionately describe as Neanderthal protein design, because it's taking what exists already and making small changes. Okay, so I thought, since I was speaking in the same session as Floyd, to show what we can do when we can start incorporating unnatural amino acids. So on the computer, unnatural amino acids and backbones are really no different from natural ones. We can model them in the same way. So we can actually, using combinations of L and D amino acids, the natural amino acids and the mirror images, we can design a whole, a very huge array of new types of molecules which don't exist in nature. And what I'm showing on this slide are just short macrocyclic peptides, where the structures, our designs are in green, and then the structures here are in gray. These molecules can have very interesting properties. They get across biological membranes very effectively. And so this is a positive control for getting across. This is a standard Transwell assay for measuring how well compounds get across membranes. And you can see all of these compounds shown here are traversing this cell layer very, very effectively. What's exciting is so we can design these things, and not only do they get across membranes, but we can design them very, very accurately. So this just shows crystal structures comparing our designs to that we have on the designs we make on the computer to the actual structures. You can see they really come out very close. And so now now that we can design these new compounds very accurately and get them to go across membranes, it opens up sort of new avenues towards orally available drugs. And so now what we're doing is to incorporate binding functionalities into these for targets of interest. I wanted to next talk about this design of binding problem, but I'm not going to talk about it in the context of these mini proteins. I'm going to talk about it in the context of proteins. Floyd already introduced the IL-2 and its power as in immunotherapy. And I just want to describe our approach to this problem. And so here is the IL-2 receptor. It's got a beta, a gamma, and an alpha receptor subunit. A lot of the toxicity associated with the original use of IL-2 was associated with the alpha receptor subunit. So in this case, what we've done is we've designed a completely new protein with no sequence similarity to IL-2, but that folds up in such a way as to make interactions with the beta and gamma subunit, but not with the alpha subunit. And this is a crystal structure of this protein called neoleukin bound to the receptor. And I'll just show you some biochemical data on this to show you how it works. So this first experiment, we're looking at measuring a binding to the alpha subunit. So IL-2 and various mimics of mutant forms of IL-2 bind to the alpha receptor subunit, but this design protein shows absolutely no binding. In contrast, if we look at the beta gamma receptor subunits, that's what we designed neoleukin to bind. It binds quite tightly to them, more tightly than do IL-2 or various mimics. And we're the reason I showed this example, we've designed binders to quite a wide variety of different strictly cell surface receptors. We're interested in using this to modulate signaling. This one will actually be clinical trials in humans will start next year, and so it'll be very interesting to see how de novo designed proteins behave in people. So in addition to designing proteins that bind to other proteins, we've been designing proteins that bind to small molecules. And here, for example, is a small protein we've designed that binds to the GFP chromophore. So this is GFP. It's a fairly large protein. Here's our small designed protein, and it's binding the GFP chromophore here. The crystal structure of this design is very close to the design model. So this is this is a comparison here. And when we express this protein in yeast or E. coli or mammalian cells, we get a strong green fluorescence. We're using a similar approach to design now fluorescent proteins that fluoresce, for example, in the far infrared for in-tissue imaging. And those results are looking quite promising. So far I've told you about designing soluble proteins, but we can use these same techniques to design transmembrane proteins. And the example I want to give you is de novo design of a potassium channel. So as you know, there's been a lot of discussion about what the principles are of selective ion permeation. And so we sort of took a kind of a design build approach. So here is a design protein that we created that consists of 2 concentric rings of alpha helices. It looks like nothing in nature, and it doesn't have any sequence homology to anything in nature. Once we had designed the soluble protein, we incorporated in the membrane by redesigning the outer surface so it was compatible with the membrane bilayer. And in collaboration with Bill Catterall's lab, Bill's lab found that this protein is quite selective channel for potassium. This is one expressed in insect cells, potassium over the other ions you can see here. This is a cut through of the channel, as I showed before. And to confirm that the ions were actually passing through this central channel, we mutated a residue at the entrance to cysteine, and when we chemically modify the cysteine, the conductance completely goes away. So this is quite exciting because now we can systematically design channels with different properties. We've also been able to design larger channels and pores that we're quite excited about developing for applications, for example, for single molecule sequencing. So everything I've described so far is a monomer, a small assembly. And now I want to turn to designing self-assembling systems, which we often see in biology. So the first example is a protein that's now been designed to make an interface with itself that drives formation of these long helical fibers. And it's probably a little hard for you to see, but these proteins do form long fibers when they're expressed in cells. And when Justin Coleman's lab solved the cryo-EM structures, it turned out these fibers were very close to the computational models. We can do the same thing in 2 dimensions. So now here we have 2 proteins that have been designed, a magenta protein and a green protein. And when they're designed to come together to form an extensive hexagonal lattice. And that you see here. These lattices form in cells, and these are images of material that's just taken directly from cells. But we can also make the proteins separately and mix them together, in which case they form these very beautiful large hexagonal arrays. And here we see the EM data here sort of averaged and blown up. And here the computational model is in magenta and blue. So we can design these sorts of structures very precisely. I'm not going to show you the data, but we can attach various types of signaling molecules to these components. And then when we add the arrays to cells, we can get quite dramatic responses because we get very large scale clustering of receptors. Now, both of the types of materials I've told you about so far are unbounded systems, unbounded one-dimensional fibers or 2-dimensional arrays. But we can use similar approaches to design proteins that self-assemble into closed structures like these icosahedra. And so here we have, these are 2-component systems. They're 2 proteins which are designed to come together to form these icosahedral structures. And here we have the design models, 3 different views. These are icosahedra, so they have 3-fold axes, 2-fold axes, and 5-fold axes. And the crystal structures solved in Todd Yates' lab are very, very similar. So we can design even these very complicated protein materials very precisely by de novo computational design. Now, my colleague Neil King at the Institute for Protein Design at the University of Washington, right next to me, has been using these icosahedral particles to make vaccines. And for example, he's displayed the influenza hemagglutinin on the surface of these particles. And by displaying different versions, he's been able to make vaccine candidates that give very broad responses. And those will also be going into clinical trials in the near future. More recently, Neil's been working on, has very promising results with coronavirus spike protein on these particles. And that is giving very strong responses in animals to the virus. We can also build nanoparticles out of natural building blocks, one of the components. And so in this case, what we've done is we've designed a protein which, when mixed with any antibody, causes it to assemble into a nanocage. We actually, this is an icosahedral nanocage. This is experimental data. You can see it's very close. And we have designed proteins that will assemble antibodies into octahedral structures, tetrahedral structures, and dihedral structures. And this is very interesting because we can now take any antibody that interacts with any cell surface receptor and cluster it in different ways. So we've been able to activate a number of different signaling pathways, both, for example, B cells and T cells, using as well as kill cells using death receptor antibodies using this technique. And we're quite excited about it, and we're hoping, with everything I described, if you're interested and see an application of it, let me know. We're very interested in collaborating with this stuff. If you take ACE2 or an anti-SARS-CoV-2 antibody and assemble it in these nanoparticles, you substantially increase the inhibitory, the neutralizing potential. Finally, we can package nucleic acid inside these nanoparticles. In this case, we're actually packaging the nucleic acid that encodes the two proteins components of this capsid. So the result is we get a virus-like particle so it's completely synthetic though, with a protein on the outside and the mRNA on the inside. We can introduce variation into this system, harvest the particles, and then select in various ways and go around the loop and actually evolve these completely synthetic particles. We're using this approach now to try to develop what we hope will be improved biologics delivery vehicles. And I want to turn now to designing proteins that can undergo conformational changes and carry out logic calculations. Sorry about that. So the first example I want to give we've designed pairs of proteins that interact through very intricate hydrogen bond networks. Floyd talked about base pairing. Here, this is sort of the protein analog of base pairing, where we've designed these very extensive hydrogen bond networks. You can see, comparing the designs and the crystal structure, they come out very close. We can use this hydrogen bond network thing to create the analog of sort of many different complementary oligos, but with proteins. So we can take, we've designed many dozens of these complementary pairs. We can mix them together, denature them, and when we renature them, they essentially only come back in the cognate design complex. And we can use this now to do logic operations with proteins. We can combine them in different ways in sort of the same way that DNA nanotechnology logic works. The most exciting results with these sorts of systems in cells have come from a really wonderful collaboration with Hannah El-Samad's group. Here, what we've done is to design using these hydrogen bond networks, what we call as a cage and a key. The cage has a bioactive motif in it. When the key is added, the motif is exposed and it can bind a target. And so, for example, this is binding data where we add this cage to a protein to a chip that's covered with the target, and there's no binding at all. But if we add the key, then this latch comes out and we get this dramatic activation of binding. In cells, Hannah and her student, Andrew Nguyen, basically expressed versions of this cage system where what was caged was a degron, a peptide which causes the protein that's attached to be degraded. So in this case, there's no degradation because the degron is caged. But if you add a key that opens this cage, then the protein is degraded. We have a complementary cage, which is not opened by this key, and that cage is not degraded when the key is added. You can see here. This is the key expression you're looking at. If, on the other hand, we co-express a key that opens this cage, but not that one, we lose the RFP, not the YFP. And quite a wide range of bioactive peptides have been caged in this way, and this gives a way of controlling in synthetic biology the activity of proteins post-translationally. Another example is using this system is calculations on the surface of cells for the situation where, say, you want to recognize cells that have two biomarkers, say HER2 and EGFR, but not just a single one. For example, if you want to have more precision in an anti-cancer therapy. So if we target the cage to one target, to one of these biomarkers and the key to the other, nothing happens. But if on cells that have both targets, then both the cage and the key are recruited, and we get exposure of the epitope. In this case, we're simply adding a fluorescent protein that recognizes this epitope, and you can see in a mixture of these cells, we only get labeling of those cells which have both proteins on their surface. So we can very precisely identify subpopulations of cells. And we can extend this to a case where there are 3 copies, because we can send the cage to one marker, the key to another, and then a decoy which binds the key more tightly to a third. And in that way, target cells that have 2 markers, but not a third marker. So it's A and B, not C logic. And in collaboration with Stan Riddell's group, we have shown that this can work with CAR T cells. So if we have CAR T cells that recognize this epitope, then they kill cells that have the 2 markers, but cells that have a third marker, which would be a healthy cell marker in an actual use case, do not get killed because the key is sequestered away. And just as kind of a fanciful kind of other extremes, or this isn't immediately medically useful, but Alexis Corbet has been designing completely synthetic protein motor systems. He's got a very beautiful rotary motor system shown here with an axle and a ring. And in preliminary single molecule experiments with Zeph Bryan's group, it appears that we can actually drive rotary motion of this with adding a small molecule that is hydrolyzed at an active site that's embedded it here. It's very exciting, but preliminary. We've been recruiting, trying to get the general public involved in this design work. So we have this game called Fold It, where anyone can download it and start designing proteins. And Fold It players have been designing all sorts of proteins. We've experimentally tested a number of them, and it's kind of amazing. Fold It players can now design proteins completely from scratch. This shows crystal structures of some of them. And more recently, Fold It players have been designing COVID binders. And just in my last few slides, I wanted to mention what we've been doing, really focused on the last 4 months. I already mentioned my colleague Neil King, by putting the COVID, the RBD of the spike protein on the nanoparticles, has made what look to be very, very promising COVID candidate vaccines. And what we've been working on, I'll describe briefly on 2 slides, therapeutics and diagnostics. This is a diagnostic that we've designed that using that locker system, the switch system, where in this case, the switch is activated, this particular one, by anti-coronavirus N protein antibody. So you can see there's no signal, and then you add the antibody and there's a dramatic increase. So we've been using the protein-protein interface design methods I described to design small proteins that bind tightly to the spike protein. This is ACE2 bound to the RBD. This is a small design protein. These proteins are super stable. We can make them in bacteria. They're much smaller than antibodies. And we can design quite a few of them that bind in different ways. So these proteins are extremely potent. The IC50s are about 11 picomolar of our best ones. And they're about 10-fold more potent on a mass per mass basis than the best antibodies that have been described. So this is live virus and Vero cells. This is work by Brett Case and Mike Diamond's group at St. Louis. So we're very excited about these. They'll be going into animals, hamsters, mice, and ferrets soon. And so we're hoping they'll contribute to the fight against the virus. Here is, we've used that same binding protein I just described. It's 55 residues. Here's another picture of it. This is the one that has a 11 picomolar IC50 bound to the RBD. We've incorporated those into these locker sensors. So here's a picture of the sensor. When the virus is present, this is pulled out to bind the virus, which leads to reconstitution of luciferase activity. And you can see we get this very dramatic enhancement of signal in the presence of the virus. So it's about a 17-fold increase in, this is a luminescent sensor. So it's got a limit of detection right now of 15 picomolar, which is a little bit high for a protein-based coronavirus sensor, but we're hoping to improve that and also be able to deploy that in the fight against the virus. So in conclusion, I hope I've shown you we've understand the fundamentals of protein folding and assembly to the extent that we can design a wide range of proteins from scratch, and we can now actually build proteins that can be useful. Let's see, I talked about the work of a lot of people. Brian Koepnick really led the folded work. Parisa and Gaurav really pioneered or developed those membrane permeable cyclic peptides I described at the beginning. The COVID work is done by Longjin Cao and Brian Coventry and Ina, who did the therapeutic work, and Alfredo and Andy did the switch design work. The design of the potassium channel work is out of Chunfo Zhu and Peilong Liu, working with Bill Catterall. I talked about the heterodimers of Zibo Chen, the 1D fibers of Hao Shen, and the self-assembly. The 1D self-assembly was work of Ariel. How the 2D self-assembly is work of Ariel and Jacob, together with Neil and Will and Robbie designed the icosahedra, and Alexei designed the molecular machines. And we've had many great collaborators involved in all of this work. I just want to call out again Anna and Andrew, who did the great work with the locker. And yeah, thanks for your attention.
Thank you so much, Dr. Baker, for a wonderful presentation. I do see Dr. Stambrook on. I don't know if you wanted to say a few words, Peter, if you have time. If not, we can certainly open for questions, which I will turn over to Justin Keeney for moderation. And please, as I mentioned before, you can enter via the chat or the Q&A, and we can pass those along to the speakers for you.
So while we wait for questions to come in, I do have a question for Dr. Baker. Have you tested the upper limits to the amount of cages that you can, the size of the cages that you can produce? Like, I know earlier in your presentation, you were saying you could cage antibodies to help deliver them specifically to B cells and T cells, but could you make those big enough to encase a viral particle or even a small live cell?
Let's see. So for caging much larger things like cells, the best bet are those two-dimensional sheets that I showed that we can actually get to wrap around. We can actually grow them so they sort of wrap all the way around cells. The icosahedral particles have a very defined geometry, so they're better for encapsulating RNA or DNA or proteins. If you want to encapsulate something larger, then something that's more flexible and shape matching would probably be better.
Thank you.
But yeah, we're thinking about things like, could we encapsulate islet cells, beta cells with these sheets and render them non-immunogenic, for example.
So far, that seems like the only question that I've been able to see.
Same. So as a reminder, if anybody has any follow-up questions from this event, you can certainly pass them along to me at jhoma@sebm.org, and I'll send a follow-up email after this event, and we can pass those along to the presenters today. And unless we have any other questions, which I'm not seeing right now. Oh, I'm sorry. Here come a few. I don't want to cut us off too early. Here they come. Go ahead.
We just got one from Elliot for Dr. Baker. Are there any immunogenicity concerns with using de novo proteins in human systems?
Yeah, I think immunogenicity is one of the really big concerns. We've looked hard for immunogenicity in the in animals, so we haven't seen anything really all the way up to NHPs. We think it's because they're very small and very stable, so they have a difficulty getting taken up by dendritic cells and then presented. But that of course does not mean that that they wouldn't be immunogenic in humans. So as I said, we're going to be getting feedback really within the next year on that, and that's we'll have to wait and see. It's always hard to predict what how the human immune system will interact with foreign proteins. But there is one point that's worth making, that if there is an immune response, that immune response will not cross-react with a naturally occurring protein, because these designed ones are so different. So that's an advantage of building something that's as a drug that's completely different than any human protein. If you do elicit an immune response, it's not going to cross-react with the host protein, which could be catastrophic.
I'm not seeing any other questions at the moment.
Okay, well, pending any other inquiries before we sign off, I would love to thank both of today's speakers, Dr. Baker, Dr. Wilmsberg, and our moderator, Justin, for taking the time. And as a reminder, this will be recorded, it'll be hosted live on our website, as well as it'll continue to live on the Facebook page of SEBM if you want to access it that way. Please also keep in mind we have two more synthetic biology webinars coming up. One is next week on the 25th. The following and final in the series will be September 1st, I believe, the Tuesday. And we'll certainly send more information about that via email and on our social media channel. So if you enjoyed today's event, please join us next time as well. Thank you all so much for joining us today, and we'll hope to see you in the future.
Thanks.
Take care, guys.